Good afternoon, everyone. I would like to welcome you to this neuroscience webinar, jointly organized by IATRIS, Neuratrice, and the French Glial Cell Club. Today, we are very pleased to have 3 lecturers from the French Glial Cell Club speaking about innovating therapies for glial cells-related diseases. My name is Laurent Duquesne. I am project manager at Neuratrice and training manager at IATRIS, and I will be hosting this webinar today. I would like to make you aware that this webinar is being recorded, including the questions and answers at the end of this session. It will be made available on IATRIS, Neuratrice, and the Glial Cell Club website shortly after this session. We have reserved some time at the end of each presentation for questions. We invite you to write down your questions in the question box in the control panel, as you can see on this slide. Before we introduce our speakers, we will briefly present the 3 research organizations bringing this webinar today. IATRIS is the European infrastructure for translational medicine. IATRIS helps you de-risk and add value to your drug, vaccine, or diagnostic development program. We do this by providing fast, tailored access to cutting-edge enabling technologies in translational research. Via our central, you can access the vast array of clinical expertise and high-end facilities that are available within the more than 100 top-tier academic centers across Europe. Always keeping the patient at the heart of our interests, IATRIS relies on 3 pillars to help early decision-making: biological insight, validated analytical tools, and clinical observation. IATRIS' strategic research agenda follows 2 main directions. From the technical perspective, we help select and validate next-generation translational tools and biology. From the operational perspective, we foster new collaboration and encourage new funding models. You can find out more on our website, iatris.eu. Neuratrice is the French node of IATRIS. It is also a national research infrastructure dedicated to neuroscience and more precisely to the neurodegenerative diseases that are Alzheimer's, Parkinson's, Huntington's diseases, and multiple sclerosis. Neuratrice is composed of expert teams from 6 renowned hospitals and research centers, accessible through a one-stop shop. This consortium brings together all the needed bricks to build a full translational neuroscience project and gives you an easy access to multidisciplinary expertise and high-level platforms to achieve your project in neuroscience from bench to bedside. All this information and more can be found on our website. Should you have any question about Neuratrice, please feel free to contact me or our business development manager, Ali. We will be happy to answer them or to put you in contact with the experts you need to define your project. I will now hand over to Mesma, President of the French Glial Cells Club.
Well, thank you, Laurent. And so I will give a brief introduction of the French Glial Cell Club. So the French Glial Cell Club was created in early 90s to promote research on glia. Our scientific board is currently composed of 14 members across France who have different expertise on glial cells. So the main goal of our club is to gather the community of researchers and students working on glia, astrocyte, microglia, oligodendrocyte, Schwann cells, stem cell, and tanycyte, and many others, to organize scientific and social events on glial cells to promote scientific collaboration. So I just mentioned that we are organizing annual meeting, and our next annual meeting will be in south of France in October 21, near Sète. So we are also involved in the organization of a European meeting on glial cell in health and disease. Next year, this meeting will be in Marseille in early July. So we organized workshops, symposia, And also our mission is to provide travel grant to students and young researchers to help them to attend to this scientific meeting. The other goal is to raise awareness of the general public on glial cell, and more information about our club and our activity can be found on our new website, frenchglialcellclub.fr. And you can also follow us on Twitter. I will now hand over to Olivier Pasquale for the presentation of our speaker today, and I thank you all for attending this webinar.
Hello, everybody. So our first speaker today will be Dr. Caroline Sevin. Caroline is a neurologist and pediatrician at Bicêtre Hospital and a member of the gene therapy team led by Nathalie Cartier at the Paris Brain Institute. She develops gene therapies for some leukodystrophies and is vice president of the Scientific Council of the European Leukodystrophy Association. She is one of the managers of the Reference Center for Rare Leukodystrophies and Leukoencephalitis. The second speaker will be Dr. Piotr Topilko from Biomédicale Research Institute Mondor. Piotr is an internationally recognized expert in the field of Schwann cells biology on neurofibromatosis, type 1, also called Nf1. He leads outstanding research on Schwann cells, neural crest-derived boundary cap cells, also called BC, and Nf1. He identified BCs as cells at the origin of the neurofibromas in Nf1. His team recently set up Nf1 mouse models, which has led to substantial progress in the understanding of development of neurofibroma, including their malignant transformation, and those models are used for drug screening studies. Our third and last speaker will be Dr. Nicola Trico. Nicolas is team leader at the Institut des Neurosciences de Montpellier. Before, he has been working at the Swiss Federal Polytechnic School of Switzerland and at the Memorial Sloan Kettering Institute in New York City in the US. He is an expert in the biology of myelin and peripheral nerves, winner of the ERC Consolidator Grant in 2012. He develops therapeutic approaches for disease of the nerve and for neurodegenerative disease linked to myelin in the brain and spinal cord. I will now give the floor to Caroline Sevin, our first speaker.
Good afternoon, and thanks to the organizer for inviting me to participate to this webinar. And I would like during this presentation to try to expose you some of the challenges and the first clinical results in gene therapy for leukodystrophies. Leukodystrophies are a large group of genetic disorders affecting CNS white matter, myelin and oligodendrocytes, and up to now more than 60 different diseases have been described, and this number is growing very, very quickly. And the definition itself of leukodystrophy has a bit changed over the time. First focusing on the myelin and oligodendrocytes, and now including as a cause some defect in other CNS cells, particularly astrocytes and the microglia cells. And this leads to a new recent classification that is based according to the primary cell defects, either oligodendrocytes with hypomyelinating, demyelinating, or vacuolizating leukodystrophy. You have in brackets some example of this disease, but also astrocytopathy like Cash or Alexander disease, or microgliopathy like CSF1R disease, and finally also leukoaxonopathy and involvement of brain vessels. In terms of clinical presentation, the abnormal myelin leads to axonal dysfunction And this is the cause of the clinical symptoms. And the most classical symptoms are cerebellospastic syndromes associated or not with extrapyramidal disorders. And there is a cognitive impairment that develops more or less in patients, and it's a hallmark of the disease in adults, but also in children, but particularly in adults. And we should notice that in some of these diseases, daily life events may trigger either the onset of the disease, I mean the clinical onset, or degradation on the acute phase of the disease. And this, these events are like infection or head injury. And we have lot of patients who, in whom the disease was diagnosed following infection or following an injury. We must also look at other symptoms, neurological symptoms or extra neurological symptoms that may help for the diagnosis. And typically there is two kinds of evolution. The first one is a developmental presentation, meaning that the disease is present as soon as birth or first week or months of life, with a relatively slow progression of the disease, and even sometimes the child or the patient may have some improvement of some progress. And this is the case in hypomyelinating disease, leukodystrophy, whereas in demyelinating leukodystrophy, there is most often a period free of symptoms, and then sort of kind of stagnation, and then very rapid degradation and degradation, motor and in the motor and cognitive fields. When we have this kind of clinical symptoms, of course, we perform a brain MRI, and brain MRI may also allow to differentiate between hypo and demyelinating disease. And in the group of demyelinating disease, we can have some typical features in some of these diseases, like you can see on MLD, metachromatic leukodystrophy, or Canavan or Krabbe disease with a diffuse high demyelination. Or what you can see in adrenal leukodystrophy with a typical inflammation, which is an important part of the disease, or on the right panel in astrocytopathy, where there is a very important destruction of the myelin. So based on the MRI, we can also try to make the diagnosis. And after, of course, we go through biochemical or genetic dosing. In terms of pathophysiology and the evolution of the disease, there are three things to consider. The first one is, of course, there is always a primary defect due to the genetic defects, but this defect may involve a secreted enzyme, a membrane protein or another protein. And it's important, of course, to, when you talk about gene therapy, to be able to determine which cell types you will have to target: oligodendrocyte, astrocyte, microglia, and so on. The second thing is that following this primary defect and this biochemical consequence, there are a lot of secondary mechanisms which are not specific of each disease and which are involved, for example, neuroinflammation. Neuroinflammation is very, very important in terms of pathophysiology in leukodystrophy, and also other secondary deficiency, including impaired mitochondrial function, oxidative stress, abnormalities in lipid metabolism, autophagy, apoptosis, and so on. And this raises the second question, which is maybe it will not be sufficient just to replace a gene in such a brain which has a lot of secondary mechanisms. And it's very important to think about that when you develop a gene therapy approach. The third question, of course, is when will we be able to do this treatment when we speak about gene therapy or any therapeutic option, of course. As I said, some diseases are already present at birth. For other diseases, there is a therapeutic window free of symptoms before the disease occurs. There are some diseases with a very rapid evolution, some other diseases with a progressive evolution. And for each of these diseases, of course, we will have to choose when we must treat to be efficient and when it will not be possible to treat. Of course, the earliest is the best. The most efficient, of course, would be probably at a pre-symptomatic stage. As I said, what we can do now, we can, if we come before the symptoms, we can try to prevent or protect the patient from the disease, and probably it's the most simple. If we are at the phase of the first symptoms, so when the disease is not progressing so quickly, we can try to slow down or even correct. But when we are at the phase of the very rapid degradation, when there is a lot of destruction in the brain, of course, we must say that today we cannot repair or replace. So it's important to know that when we start a treatment, if we treat patients with no myelin and no neurons, it will not work, of course. There are a lot of therapeutic options, including in the red circle, in vivo gene therapy and hematopoietic stem cell gene therapy. As you can see, there are several kind of treatments, either based on acting on the primary defect, but also on the secondary mechanism. And we can, based on what I said a few minutes ago, we can anticipate that probably in this patient and in this very severe and complex disease, we will have to combine treatments to be sure to be efficient. Now I will focus on gene therapies for leukodystrophy. Two different gene therapy strategies have been developed and have entered in the clinics. The first one is ex vivo gene therapy using hematopoietic stem cell gene therapy with a lentiviral vector. This strategy is under evaluation in adrenal leukodystrophy and metachromatic leukodystrophy. And the second option is in vivo gene therapy. It means direct administration of the vector into the brain using adeno-associated vectors. And this has been evaluated in metachromatic leukodystrophy. Both these procedures, of course, have some advantage and disadvantage. And once again, we will have to choose based on what we want to do, in which disease, in which patient, in which rapidity. We will have to choose between these two options. For in vivo gene therapy, as I said, AAV vector are the most used. These vectors transduce mostly neurons, but we can modify that based on changing the serotype of the vector. There are some serotypes that may transduce more other cells than neurons, and also based on the choice of the promoter. We can use different routes of administration, directly into the brain or into the CSF, or now, thanks to new serotypes that are able to cross the blood-brain barrier, we can also anticipate that we are able and we will be able to administer the vector intravenously, which is, of course, less invasive. We will have to think about the rationale to do or not immunosuppressive therapy, because of course this vector, even if they are not very immunogenic, they can lead to an immune response against the vector itself, but also against the transgene. And probably that, particularly when we use intravenous administration, we will have to think about that. One of the most positive characteristic of this approach is that it's had a very rapid effect. In a few weeks, 2 or 3 weeks, there is a maximal expression of the therapeutic gene, and it's very important if you have to treat patients with very rapidly progressing disease. The mechanism, of course, you correct some cells, but not all the cells. Small percentage of cells, probably, particularly in humans. And in some disease, not all, but in some disease, for example, in lysosomal disease, the transduced cell is able to secrete the enzyme, and this enzyme may be endocytosed by adjacent cells, and this may increase, of course, the efficacy of the procedure. So it's an important phenomenon, but it's not true for all the diseases. In hematopoietic stem cell transplant, gene therapy, in fact, you take the hematopoietic cell of the patient, and then you transduce them ex vivo with a lentiviral vector, and you re-administer the treatment, the cells to the patient after intensive myeloablation. It's like an autotransplantation of corrected cells. This mechanism is based on the fact that the cells that you have re-administered, the transplanted cell, one part of these cells are able to go into the brain and differentiate in a microglia or brain macrophages and just replace or substitute to the deficient cells. That's the main mechanism. And in addition, of course, you may also have the cross-correction mechanism for lysosomal enzyme or secreted enzyme. One problem with this treatment is that, in addition to, of course, the fact that it's not a very, it's relatively invasive because of the myeloablation and so and so, but it's also a treatment with a delayed effect because it takes months before the global turnover of the microglia into the brain, and so it takes about 9 to 12 months to be completely efficient. So you can just know that if you have a disease with a very rapid progression, this treatment may not be efficient because it will be too late. We will now speak about two diseases, adrenal leukodystrophy and metachromatic leukodystrophy, for which there are clinical trials using ex vivo and in vivo gene therapy. First disease is adrenal leukodystrophy. This is the most frequent leukodystrophy in men. It's an X-linked metabolic disease. And there are two forms of the disease, the adult forms, which is named adrenomyeloneuropathy, which is a spastic paraparesia, but without cerebral involvement. And the cerebral adrenoleukodystrophy, which is a very, very rapidly progressing and inflammatory leukodystrophy, occurring most often in boys, in children, but also in adults. A few months later, the child starts to have some more important symptoms with neurological, motor, and cognitive symptoms. And at this time, when we make the diagnosis, it's too late because the lesions are already too advanced. And then there is a very, very rapid degradation with vegetative states and severe spastic quadriparesis vegetative states in a few months and premature death. So it's a terrible, terrible disease. And unfortunately, if we don't treat sufficiently early, we cannot have any effect. And the treatment is allogeneic hematopoietic stem cell transplant, which is performed in patients for several decades. And if you do this treatment at a very early stage of the disease, it means when you have already a lesion, of course, you must have a brain lesion to do this treatment. You must have some sign of inflammation in the brain. And at this time, for most of them, there is no symptoms. And if you do the transplant at this time, you are able to arrest or avoid the disease. However, allogeneic hematopoietic stem cell transplant is associated with complication, including, of course, mortality, around 10%, graft failure, and also GVHD, graft versus host disease, which is a very important problem and which requires long-term immunosuppression in the patients. That's the reason why, because of this complication, because also of the fact that you don't find a donor for the patients, that Patrick Aubour and Nathalie Cartier have developed around 15 to 18 years ago a gene therapy, an ex vivo gene therapy approach for adrenoleukodystrophy using an antiviral vector. And they have first demonstrated that this approach was able to globally have the same results than allogeneic hematopoietic stem cell transplant in 4 patients affected with childhood cerebral ALD. And then there was a more phase 2-3 trial that for which I will give you some result that was developed by Bluebird Bio, including patients with cerebral ALD, but a very, very beginning of the symptoms with a mild demyelination with no motor symptoms and evidence of active cerebral ALD, I mean inflammatory disease. And they have enrolled around 30 patients. You can see here the characteristics of the patients. Mean age was 6 years. That's what I was saying when I described the clinical symptoms. As you can see, the low score, which describes the demyelination, is very low, median range of 2. This score goes from 0 to 35 when there is a severe demyelination. And for the neurologic function score, the NfS score, globally no or minimal symptoms. And what we observed was finally, as we can say, exactly the same results than for allogenic hematopoietic stem cell transplants. In the patient, you can see on the upper left panel that most of the patients remain asymptomatic in terms of neurologic function, except one patient that you can see with an increase in the score. This patient was unfortunately a French patient, but was treated too late. I mean, and it deteriorated. It's not due to the treatment. Unfortunately, he was just treated too late. When you look at the MRI severity score of the demyelination score on the upper right panel, you can see that, of course, as I said, it takes some months to be efficient. So in the first months after the treatment, you have some increase in the demyelination, in the demyelinating score. But after 9 to 12 months, there is a stabilization with relatively low demyelinating score. Of course, and it's of importance, the treatment was able to stop the inflammatory process. As you can see in the lower left panel, where you can see on the picture the white lesions that are lesions, inflammatory lesions, and that disappear 9 to 12 months after injection. So, and the last thing was there was no complication with this treatment, no graft, of course, no graft failure, and of course no GVHD because it's own cells of the patients. No insertional oncogenesis because an antiviral vector is a virus that which DNA insert into the genome, so there may be some risk. And there was a safety profile, and clearly now this hematopoietic ex vivo gene therapy is, I hope will soon be available to treat patients. The treatment is under evaluation at this time, and probably we will be able to treat patients later this year, I hope so. And we will have to choose between a conventional allogeneic hematopoietic stem cell transplant and gene therapy. It will not be easy, but probably that gene therapy would be preferred when there will not be any intrafamilial donor. There's two forms of the disease: an early onset form, which with first symptoms before 4 years, and which is very homogeneous with patients who initially develop normally, then there is a sort of stagnation, and then a very, very rapid, in a few weeks or months, a very rapid degradation, cognitive and motor degradation, leading to vegetative state and premature death. For this form of the disease, there is no treatment when the patients are asymptomatic, and if pre-symptomatic patients, we will see on the next slide that we have tried to treat them by allogenic hematopoietic stem cell transplant. And in the late onset forms, with onset after 6 years, these forms are more heterogeneous, and in terms of age at onset, evolution, and severity, they are more progressive, and when treated sufficiently early, they can be improved, but not cured by hematopoietic stem cell transplants. And then, as I said, we have treated the patients with a pre-symptomatic early onset form of the disease. I mean, we've treated 3 patients at 2 months, 3 months, and 9 months. And if I give this result, it's because it was very interesting, because we were able, with this treatment, to stop the demyelination. It means that the patient do not develop demyelination. However, they develop a severe disease, a severe motor and cognitive disease, more progressive. And finally, 2 out of the 3 patients died around, 10 to 12 months, which is later than the natural history, but they were very severely affected. And it means that in addition to the leukodystrophy, probably there are other mechanisms that the transplant may not target. However, in this disease, it's one of the diseases for which there are more therapeutic approaches. There are currently 3 approaches in clinical trials. The first one, I will not speak about it. It's enzyme replacement therapy using intrathecal administration into the CSF. And the two other options are intracerebral in vivo and ex vivo gene therapy. For in vivo gene therapy, we have developed with Patrick Aubour and Nathalie Cartier clinical protocol to treat patients with intracerebral administration of an AAV10 vector directly into the brain at 12 injection sites. And this trial was conducted in about 7 or 5 years ago, 7 years ago. And we have treated 4 patients. These patients were treated with 2 doses of vector, one 10 to the 12 and 4 10 to the 12 vector genome without immunosuppression. And we have enrolled 4 patients. 2 were the inclusion criteria were patients with early onset forms of MLD. It means onset before 5 and at a very early symptomatic or pre-symptomatic stage. We have treated 4 patients, and unfortunately, what we can observe that even if we are able to increase our activity into the CSF, meaning that the vector was efficient, was functional, we were not able to have any improvement in the patient that developed exactly the same disease than the natural history, and particularly the pre-symptomatic patients. So it was very negative results. But for this reason, we stopped the trial. And the other trial that is developed in MLD and is currently under evaluation is ex vivo gene therapy, and it's a treatment that is developed in Milano by Orcar Therapeutics in pre-symptomatic or early symptomatic patients with early onset metachromatic leukodystrophy. And they have treated more than 20 patients, and no more than that. And what they observed is that first they are able to restore normal activity in the blood and in the CSF. And these are the results, the clinical results are very, very impressive, because if you treat the patient at a pre-symptomatic stage, for most of the patients, you are able to prevent the motor and cognitive impairment as well as the demyelination. This treatment is very, very important for the disease, and now it's like in ALD, under evaluation for clinical use, and we will be able to treat patients probably in the next year. So in summary, what we can say that leukodystrophy are good candidate for gene therapy approach. Of course, it's a monogenic disease, a very, very good candidate. But we must know more probably about the pathophysiology and natural history of each disease, because if we don't know that, we will not be able to target the right cell at the right time. Additionally, we will have probably to combine treatments, and particularly for symptomatic patients, because the promising results that have been obtained using ex vivo gene therapy in ALD and MLD are in very, very early symptomatic patients. But it's not the patient that we see in the clinics. Most often we see patients who are already symptomatic. So we will have to combine treatment, and probably that we will have to combine a treatment with a rapid action, for example, in vivo gene therapy. Now we can probably administer the treatment intravenously, and we probably will have also to combine in most of these diseases, maybe hematopoietic stem cell without gene therapy, because there are additional effects due to the transplantation itself, due to the reset of the immunity and due to the correction of the microglia into the brain. So I think that the future probably is around that, and probably also to add some treatment against secondary mechanism. So that's all. I finished, and sorry if I was late, because thank you very much.
Thank you. He was asking about the use of AAV versus hematopoietic stem cell. Which strategy to use depending on a type of leukodystrophy? What is the rationale of using one or the other?
I think that, of course, it depends on which strategy you are working in your lab. That's not the answer, of course, but it's right. It means if you are specialized in AAV, maybe you will prefer to try to do in vivo gene therapy. If you have specialized in hematopoietic stem cell gene therapy, that's the most simple answer, but it's not the answer. To my opinion, I think that now we have developed gene therapy, in vivo gene therapy for MLD with intracerebral injection, and it was completely negative as a result. So I think that, and that's the reason why I tried to explain that in the last slide. I think that it will not be sufficient in any case for the disease, maybe not for all leukodystrophy, but for ALD, for MLD, also for Krabbe disease, which probably is the same kind of mechanism. In most of these diseases, there is a part for an inflammatory process into the brain, neuroinflammation, and probably that microglia in some of these diseases is not completely functional or is injured by the disease. So I think that at the moment maybe you will have to do transplant. I think that's mandatory. It's not sufficient maybe, but it's mandatory. And using in vivo gene therapy with AAV, you will have a rapid action, because the problem of the transplant is that it's a long-term action. So if you combine something with a very rapid action and something with a mechanism that involves correction or modification of the immunity into the brain, maybe you will have a good results. So it doesn't answer the question, but I think that in my opinion, and that's what we will develop for metachromatic leukodystrophy, is to associate in vivo gene therapy with a rapid action and directly into the brain or intravenously or not, and also transplant to act against this part of the disease, which is neuroinflammation, and which is not so easy to understand.
Okay, thank you.
Okay. Okay, so good morning, everybody. And again, I would like to thank the organizers, and especially my colleague and friend Nicola, for giving me the opportunity to participate in this short symposium. So I'm Piotr Topilko, and I'm heading research team at Institut Mondor of Biomedical Research at Créteil. And today I will focus my presentation on the efficacy of MEK inhibitors to treat neurofibromas in Nf1 type 1 Nf1 patients. So probably some of you already know this disease, because it's curiously is one of the most frequent rare genetic disease, also called Recklinghausen disease. So it's a simple and complex disease, because from genetical point of view, this disease involves only mutations in only one gene, a lot of mutation covering the whole coding sequence. But from the pathophysiological aspect, it's a very complex disease, because as you can appreciate on this slide, patients develop plethora of neuronal and non-neuronal symptoms. So Nf1 affect more or less 1 in 3,000 people worldwide, and is caused by biallelic inactivation of Nf1. It's an enormous gene encoding a protein named neurofibromin. What is neurofibromin? It's a negative modulator of RAS activity. It's a kind of brake to RAS signaling. So as you can see on the right panel of this slide, all patients are developing what we call benign cutaneous, benign nerve sheath tumors in the skin, at the skin level, which we call cutaneous neurofibromas. Some of them can develop hundreds or thousands. So as I mentioned, so all patients will develop at puberty and beyond nerve sheath tumors, which are totally benign, never transform in the skin. However, about 30% of patients will develop at prenatal stage or early postnatally plexiform neurofibromas. Those are also benign tumors affecting deep nerves bundles, innervating often vital organs. And finally, some of those plexiform neurofibromas, which we expect to about 10 to 15%, will undergo a spontaneous transformation into extremely malignant peripheral nerve tumors shown on the right. I don't know, should I return to the second slide? So very briefly, to make the long story short, so so all patients will develop what we call benign nerve tumors at the level of nerve terminals in the skin. We call them cutaneous neurofibromas. Those tumors emerge around puberty and beyond, but never transform into malignant tumors. In addition, about 30% of NF1 patients will develop what we call plexiform neurofibromas. They develop along the larger nerves in the soft tissues, and about 10% or 15% of them will spontaneously transform into extremely malignant neurofibrosarcoma, called also malignant peripheral nerve sheath tumors. So I also mentioned that those tumors are caused by biallelic inactivation of NF1 gene in peripheral glia called Schwann cells. There is no efficient and permanent treatment to prevent or slow down development of those tumors. And finally, as you can see on the left part of the slide, some patients will develop additional non-neuronal symptoms as café au lait macules. Those are aggregates of melanocytes, bone and eye lesions as pseudoarthrosis and scoliosis, and also optic glioma. And the list of those non-neuronal symptoms is much longer. So as I already mentioned, NF1 is a negative modulator of RAS activity, as you can see on these drawings. So it's a phosphatase, so NF1 will dephosphorylate RAS. And so when we knock out NF1, RAS is hyperphosphorylated and will activate several signaling pathways, including the RAF, MEK, ERK1, and ERK2 pathway, which was probably the best characterized in NF1 patients. So NF1 neurofibromas are complex tumors that contain, as I mentioned, biallelic NF1 loss in Schwann cells, but there are other cells present in the tumor as fibroblasts that secrete an important amount of collagen, macrophages, mastocytes, and those tumors are highly vascularized. Of course, all other cell types except Schwann cells are heterozygous for NF1. So, so the first essay to slow down the RAF, MEK, ERK signaling pathway due to NF1 mutation were performed by using inhibitors, pharmacological inhibitors of ERK1, ERK2 phosphorylation. I just showed here two molecules, selumetinib and binimetinib, but there are more than that, which have been used both mostly on mouse models to explore the efficacy of this approach. So first, to make the long story short, in the first preclinical trial, the researchers used selumetinib and on animal model recapitulating plexiform neurofibromas called desert hedgehog NF1 mice. As you can see, those mice develop large plexiform neurofibromas, mostly at the level of nerve roots, but also along the deep nerves and the soft tissues. And so here you have the results. They treat twice a day with a dose of 10 milligrams per kilo by oral gavage, 5 days a week over 8 weeks. And they observe a very significant significant shrinking in the size of those tumors by using this experimental paradigm. So this was the a pioneer studies to perform the first clinical trial with the same MEK inhibitor, selumetinib, on 24 young patients with the inoperable plexiform neurofibromas. So here, they used doses from 20 to 30 milligrams per square meter. Before this study, 20 milligram was the maximum tolerated dose described in the literature, and the children were treated by the kind of cycles of 28 days. And here you have a number of cycles. And the results are present here. So some of the patients responded very efficiently, and the tumors shrink, as you can see on this picture. After here, the month of treatment I indicated. However, the penetrance was not very high because some patients do not respond efficiently to this treatment. Finally, so you have the examples of the children treated in this trial before and after treatment with selumetinib. So another problem is that the use of prolonged use of selumetinib induce numerous side effects, and I just mentioned those two, but the list is much longer, as bleeding, urticaire, and there are some eye and problems and so on. So this is probably the main reason why the trials was performed only on children with the inoperable plexiform neurofibromas, so located in the regions which cannot be surgically treated. So finally, as shown here, when they stopped treatment with selumetinib, in some children they observed slow regrowth of those tumors, so the shrinking might be transient and not permanent. Okay, so and in this trial, the efficacy of treatment on cutaneous neurofibromas present in all Nf1 patients couldn't be explored because, as I mentioned in my introduction, cutaneous neurofibromas develop at puberty and beyond, so they couldn't address this question in this trial. For this, unfortunately, at that time, there were no animal models recapitulating development of cutaneous neurofibroma, so it was an urgent need to build such model. This is exactly what we have done. So a brief short introduction. So we came from a basic research in the field of Schwann cells. So a long time ago, we identified a particular type of glial cells, which sits at the interface between peripheral and central nervous system, which we call boundary cap cells. And what we showed is that these cells, the red cells, will crawl, migrate along peripheral nerves into the periphery, and they will colonize the nerve roots and then nerve terminals in the skin, as shown here. Here you see the genetic fate mapping of boundary cap cells in red. You see they colonize the nerve roots, dorsal root ganglia, and then they crawl along the spinal nerve to sit finally at the level of nerve terminals. In both locations, they will give rise to a various type of Schwann cells. And if you remember, those are two locations where neurofibromas develop with a higher incidence in Nf1 patients. So this is the main reason why we suggest that boundary cap cells or their glial derivatives might be at the origin, at least, of some of those neurofibromas. So to explore this hypothesis, we built the model, mouse model, in which we target Nf1 loss specifically in boundary cap cells and their derivatives. And as you can appreciate on this slide, mutant mice for the first time develop all type of neurofibromas, including paraspinal, plexiform neurofibromas, subcutaneous plexiform neurofibromas, cutaneous neurofibromas, those are those red aggregates. And finally, and very unexpectedly, for the first time, we observe that in some of those mutants, subcutaneous plexiform neurofibromas undergo spontaneously transformation into malignant peripheral nerve sheath tumors. So in my opinion, we have a really unique model that faithfully recapitulates different aspects of neurofibromas development as described in patients. Well, we made, by analyzing those animals, we made another very interesting observation. So since mutations were performed during mid-gestation in the boundary capsules, we analyzed the young skin of those animals before development of fully blown cutaneous neurofibromas. And as you can appreciate here, we observe the presence of a kind of microtumors with the much younger animals already at 6 months. And those tumors will spontaneously progress, slowly progress, to give rise to cutaneous neurofibromas at 1 year and beyond. But what we discover is that skin trauma tremendously accelerate this maturation of microcutaneous neurofibromas into cutaneous tumors, which first suggests that skin trauma and inflammation might play a very important role in development of cutaneous neurofibromas. And second, by doing so, we set up a kind of inducible model of cutaneous neurofibromas, which will be perfectly well adapted to start the first trials. This is exactly what we have done. So we address the question, what is the efficacy of MEK inhibitor to prevent or block development of cutaneous neurofibromas? So we performed 2 trials, first called systemic. So we applied binimetinib by using the same concentration as in a preclinical trial with selumetinib. And so we treat those mice from between 6 and 7 months, where one administration per day, 5 days a week during 1 month. And then we sacrifice those animals and we analyze the skin and the spinal cord. And the results are presented here. Animals treated with placebo, you still in the skin observe numerous traced cells that express high level of phospho-ERK. In those animals, we observe paraspinal plexiform neurofibromas, and in stark contrast, in animals treated with binimetinib, we observe a very significant reduction in the number of tumor cells in red, strong reduction in phospho-ERK, and there is a kind of resorption of, there is no paraspinal plexiform neurofibromas in treated mice. However, as I already mentioned, the binimetinib, so oral administration may have a prolonged oral administration, might have several side effects. So we decided to complete this study by a topical application. So here we treat the animals during the same period from 6 to 7 months, local application of the drug in a cream, and we sacrifice 50% of them at the end of treatment, whereas we keep the remaining animals for the next 6 months. Results are presented here. Animals treated with placebo, again, you observe numerous tumor cells expressing tomato, high level of phospho-ERK, and the presence of cutaneous neurofibromas at the site of treatment and outside of this area. In the animals treated with bini, again, we observe a severe reduction in the number of tumor cells correlated with the reduction in the phospho-ERK, and more interestingly, 6 months after the end of treatment, there's still no cutaneous neurofibromas in the treated areas, whereas we observe numerous cutaneous neurofibromas all around. So those results are very promising in terms of using binimetinib for treatment of cutaneous neurofibromas, at least in this experimental paradigm. So we think that a treatment with the MEK inhibitor alone might not be sufficient to to permanently shrink those tumors. So we are searching for other targets, and we got an RNR funding with our colleagues from Orléans to test a totally new target. This is a RAC-RO pathway target in which several limb kinases were described as important. And so our colleagues developed limb kinase inhibitor. And in the next year, we will use our mouse model to test the efficacy of this limb inhibitor alone. And if the results are promising, maybe in the next step in combination with the MEK inhibitors. And finally, if you remember the signaling pathway of RAS regulation, so the Nf1 is a kind of negative modulator of RAS activity, whereas the SOS and SHICT are a positive regulator of RAS activity. So now we are in negotiation with the pharmaceutical company who developed those inhibitors against SOS, and we are planning in collaboration with them to start trials again on cutaneous neurofibromas by using our mouse model, where we will treat those animals with those inhibitors separately or in a combination. Okay, so before I stop, just for the few minutes, I would like to switch to a completely different aspect of our study. So you remember in our model, we observed that some plexiform neurofibroma undergo a spontaneous transformation into malignant peripheral nerve sheath tumors. And so what we by analyzing this mechanism and those animals, we discovered that this evolution of benign into aggressive tumors passed through the intermediate stage, which we called dysplastic neurofibromas. What are dysplastic neurofibromas? It's a kind of intermediate stage, but is characterized by a massive invasion of the tumor by T lymphocytes, as shown here. Their level is low in plexiform, very high in dysplastic, and then they disappeared in the transformed tumors. So we performed a lot of work on characterization of those dysplastic tumors, including single-cell transcriptomic analysis. And the example of those studies are shown here. And what we discovered, so again, the tumor cells are here, and all the other cell types, or at least most of them, correspond to immune cells with a high infiltration by T lymphocytes. And you can see most, if not all of those T lymphocytes in this tumor, they expressed a checkpoint marker PD-1, which is extensively used to treat various type of other type of cancers to reactivate exhausted T lymphocytes. So we think that immunotherapy might be a good option to treat either dysplastic tumors before they transform into MPNST, both in our mouse models, and then later on maybe in a patient. So I will stop now and just thanks my colleagues who've been involved in this work, especially Kasia Radomska, Fanny Kulpier, and Pierre Wolkenstein, which is a clinician and he is in our team. Thank you very much.
Thank you, Piotr. Very nice presentation. So if you have question online, no. So Piotr, I have a question. Most of these inhibitors are also, the ERK pathway is also involved in myelination. So I was wondering in your strategic line, if you look at myelination after treatment with this inhibitor?
Yeah, an excellent question and a big surprise. because I didn't mention this, but this might be seen here. Let me see here. No, it's not very clear, but surprisingly, what we observe is that myelinated Schwann cells are not affected by the binimetinib trial. So there is no sign of demyelination in those cells, not at all. However, what we observe in the Nf1 myelinated Schwann cells, because myelination is not affected by the loss of Nf1, at least in our model, the level of phospho-ERK seems to be much lower comparing to the high level of phospho-ERK in unmyelinated Schwann cells, you know?
Yeah, okay. This is very interesting, yeah.
Yeah. Thank you. Do you have other questions or no?
Olivier?
Yeah, not that I can see from the chat anyway.
Okay. And one question, Piotr, is the use of a desired CRE promoter. So why not using CROX20 CRE?
Of course.
Of the origin.
Yeah, so I did mention that, but there are a plethora of mouse models carrying Nf1 loss in Schwann cell lineage. And curiously, none of those models, excepting in none of those models, the mice develop cutaneous neurofibromas. They only develop plexiform neurofibromas, excepting CROX20, because CROX20 is a bit more complicated. CROX20 is expressed in myelinated Schwann cells, but also in the boundary cap cells. So the only neurofibromas developing at the nerve roots. But as I mentioned, inactivation of Nf1 in myelinated Schwann cells have no impact on development of neurofibromas. So the authors of this work conclude, and we support this observation, that myelinating Schwann cells are not at the origin of this disease.
Okay. Okay, thank you.
Thank you.
So first I want to thank you Caroline and Piotr for the very nice presentation. So the goal of this meeting was webinar was to show that glial cells are really important, and they are beside neuron, but you see when they go wrong, then a lot of disease can occur, some really dramatic disease. And now we are able to try to cure this disease, or at least treat them. So in my case, I'm going to show you a gene therapy approach to treat Charcot-Marie-Tooth disease, peripheral nerve disease. So Charcot-Marie-Tooth disease, and in particular, it's a peripheral neuropathies that are, some of them are axonal disease. So the problem comes from the axon that don't work properly. But most of them actually are due to the myelin problem. So you know, in peripheral nerves, the axons are covered with myelin, and it accelerates the nerve conduction velocity. And while it occurs, the development of this myelin occurs late during the development, actually the myelin is critical, because if the nerve conduction is not fast enough, then you get a lot of problem, muscular problem. So it's a case, for example, for CMT1A disease, which is Charcot-Marie-Tooth 1A disease, which is the main peripheral neuropathy, hereditary peripheral neuropathy. It represents 70% of all myelin-related CMT disease. So the prevalence is 1 to 5 on 10,000. So it's a rare disease, but not that much rare. And you see that the main defect in this disease. So you see the main problem of this disease. It's a peripheral. It's a extremities problem. So you get a limb extremity problem, such as a feet and deformation that are due to the weakness of the muscle. And also you get this walking problem that is named foot drop walking or stepping, that the people are not able to lift the feet to walk correctly. So actually, this peripheral nerve disease, it's a developmental disease. So meaning that the average age of onset is 12 years. Because myelination starts late during development, it starts around birth in the humans. So you see that actually the problem comes quite early because it's developmental, late postnatal development. So the main problem that you will get is that the people that will get the disease in the most severe kind of disease, they will have to follow a lot of surgery, orthopedic surgery, in order to correct the deformation that you can see here. And so this 80% of all the people will get hand and feet deformation, and 30% of them will get EV surgery, orthopedic surgery. And so far, there is no treatment for this kind of satisfying treatment for this kind of disease. So what we develop in animals is a novel microinjection technique with pressure pulse to inject the virus inside peripheral nerves without much trauma. So you see with what I show here in animals, and you see in rat pups, we have all a setup to inject the vector into the nerves. And we use a microinjector, electrical pulse generator to produce pulses of pressure. And then we inject nanoliters, successively a lot of nanoliters in order to fill the nerve with the vector. So here we can inject up to 10 microliter in adult mice and 25 microliter in adult rat, but we can also inject newborn animals. And using this technique, we developed AAV9-based gene therapy. So what we did is inject AAV9 expressing GFP inside nerve of adult or pups, mouse and rat, and directly in the nerve. And you see what we get. So here you see the GFP in the nerve, and you see you will get a lot of transduced cells. And these cells are long and large. And when you do a CARS imaging, so CARS is for coherent anti-Raman Stokes microscopy. It's actually like a non-linear microscopy that allows you to see myelin, but without any staining. And you see, so here you see myelinated Schwann cell in the nerve. And you see that when you overlay both staining, the staining on the CARS imaging, you see that all these cells all the green cells are actually red cells. So you see all these green cells are myelinating Schwann cells. Well, we were able to confirm that because we use a marker for myelinating Schwann cells, that is the icadherin. It's only expressed in myelinating Schwann cell in the nerve. And you see that all the green cells express icadherin. The characteristic of the myelinating Schwann cell is that at the extremities here, you have a node of Ranvier. Okay, and then when you look at the expression of node of Ranvier markers, so it's sodium channel, voltage-gated sodium channels, you see that they are localized at the node, and the node is just after a green cell, showing that these green cells are myelinating Schwann cells. So we did the same in cross-section. Then we labeled the axon with TUJ1 marker, and then you label myelinating Schwann cell with MBP, myelin basic protein, and you see that all the red cells are actually green cells. So what we found, it's actually a high specificity of AAV9 for myelinating Schwann cell when it's injected directly in the nerve. So here you see, you count the number of green cells that are myelinating Schwann cell or non-myelinating Schwann cell or axon, and you see that most of the green cells are myelinating Schwann cell. Around 80–90% of the translucid cells are myelinating Schwann cell. So the vector is really specific for the myelinating Schwann cell when you inject it directly in the nerve. And then what we did is count the number of myelinating Schwann cells that were transduced. And that we did that at different places, the injection site, but also proximally and distally from the injection site. Okay? And so it's what you get in cross-section. So here you have a cross-section of a mouse nerve that has been transduced with AAV9 vector. And we also tested the AAV10 vector, which is another vector used for gene therapy in the nervous system. And we did the same in the rat, and we did the same in non-human primate, macaque. And you see that the AAV9 is much more effective to transduce myelinating Schwann cell in both mouse, rat, and non-human primate nerves, more efficient than AAV10. And then when you measure, so you count the number of myelinating Schwann cell, and you count the number of axons. And then you say for each axon, large axon, because the small one you can't see them, but the large axon, for each axon, you should get one myelinating Schwann cell. And then when you compare the number of axons and the number of green myelinating Schwann cell, you see that you get a high transduction rate. So at the injection site, the transduction rate is 93% of the myelinating Schwann cell are transduced at the injection site in adult mouse. And you see that you get a high transduction rate in almost all the species, even in non-human primate. So we could do only one non-human primate, no, only one macaque, but you see that you already get a high transduction rate without any optimization of the injection. And you see that the diffusion is quite nice because even distally or proximally, the transduction rate is also quite high. Between 70 and 90% of the myelinating Schwann cell are transduced by AAV9 when you inject in the spine, in the nerve. So just an idea about how long is proximal and distal. So basically this cover distal from to proximal to the injection site. We inject more or less in the middle of the sciatic nerve, and proximal to distal is around 2.5, 3 millimeter, 3 centimeter in the rat, for example. Okay, and you see that we get really high transduction rate over 2 to 3 centimeters. So it meaning that the diffusion rate, because there is only one injection site, the diffusion rate is quite high. So when we saw that we could do use AAV9 to transduce a lot of myelinating Schwann cell in vivo, then what we try to do is develop a gene therapy approach for CMT1A. So CMT1A is due to the duplication of PMP22 protein, and these in humans and induced myelination problem and demyelination in the nerve. And we have we use a rat model of the disease, and the rat model overexpressed the mouse PMP22 in Schwann cell, and that induced the same disease as in humans. Then you get muscle weakness, you get clumsiness during walking. Okay, they don't walk correctly, and then in the worst case you get spasticity and paraplegia. The homozygous genotype is actually lethal; they die very early, and we can only use the heterozygous mice. Actually, too much expression of mouse PMP22 is in this rat is actually lethal. So as I told you, I mean, the disease, CMT1A disease, is due to a duplication of PMP22. Okay, you get one more allele of PMP22. And that induces an excess of mRNA for the protein and mostly an excess of protein. And because the protein is like unstable, it has a 4 transmembrane domain. So most of the protein is actually misfolded, and the cell has to remove the misfolded protein. And as soon as you express too much of this protein, then you saturate the degradation system of the cell, and the cell will demyelinate. So it's a way that you lose myelin sheet all over the nerve. Okay, and then because you have less myelin sheet, you get this defect in the muscle. So the idea was to come with AV9, expressing a small hairpin shRNA targeting mouse PMP22 in the rat. So the shRNA is transformed in siRNA in the cell, and they will bind to the mRNA of the target protein, in that case mouse PMP22, and it will induce the degradation of the mRNA or block the translation of the mRNA. And that should result in less PMP22 protein. So the experimental plan was we used this vector, so U6 promoter expressing a small shRNA against PMP22. Then we use CAG promoter with expressing GFP as a reporter. We injected the animals, the rat animals, just after P6, P7 days after birth. It's because at that time myelination starts in the nerves of rats. So we treated the animal right at the beginning of myelination in order to allow a correct myelination. Because I remind you that the disease is a developmental disease, so it occurs as soon as myelination starts. So we have to prevent the disease. It's a preventive paradigm. Okay. Then we follow the animal for 12 months. So it's almost a third of the rat full life. And then we sacrifice the animal and analyze. We had used 16 animals per group. So we injected, all the animals were injected. We had one type injected with scrambled shRNA virus, then CMT1A with scrambled shRNA mouse vector, and then two different shRNA that target PMP22, but not at the same place on the mRNA. Okay, SH1 and SH2. And we injected 8 microliters, 1 to 10 on the 11 viral genome per nerve. What we found first, it's actually when you measure the amount of PMP22 that is expressed in myelin Schwann cell. So you look for a PMP22 expression over the MPG expression to normalize on myelin. You see that diseased rat, we have too much PMP22 for the same amount of myelin. And then when we treat the animal with the 2 shRNA that we design against PMP22, you see that you can decrease the expression of PMP22 relative to the myelin amount. Then what we did is look at the myelin itself. How much myelin do they have, the animals in their nerve? And we found actually, we look for MPG expression. And you see that in diseased animals, then they have less myelin that is formed in their nerve. And if you treat the animal with the vector expressing shRNA to decrease PMP22 expression, then you see that you can restore the correct amount of myelin in these rat nerves. Then when we look, first we look at the molecular aspect, then we look at the cellular aspect. And we know that in the animals, CMT1A animal, but also in CMT1A patient, the internode, so the length of the myelin Schwann cell is short. Actually, because they don't form myelin correctly, and what they form is short myelin segment. So here I represent the myelin segment. In wild type, it has to be quite long, so it's minimum 500 micrometer long, it can be 1 millimeter long. And then you see that in CMT1A patient, but also animal, they are much shorter, the internodes. And it's what we see when you look at the number of the node using CAR staining. You can see where are the nodes, and then you can measure the number of nodes per section. You see that the animals that are the animals had much more nodes on the same section, meaning that the internodes are shorter, because then you make more nodes if they are shorter. And then when we treat the animal with shRNA targeting PMP22, we have longer intervals. Then we can quantify that, and you see that we can restore the number, the density of the node of Ranvier. You decrease the density, meaning that the myelitic segments are longer. So that will help the nerve conduction velocity, because if the segments are longer, then the nerve conduction is faster. So what we wanted to check, we look for the, maybe I will jump over this one. We look for the nerve conduction velocity. So nerve conduction velocity is basically you measure how fast the axon can conduct the action potential over the distance of the nerve. Okay. So usually you see in rat the nerve conduction velocity in wild-type rat. This is in gray here. You know, it starts at 1 month around 35 meters per second, and then it's increased up to 60 meters per second. And but in diseased rat, CMT1A rat, you see that in red, that actually the nerve conduction velocity is reduced to 10 meters per second and does not increase much. Okay, and when we treat the animals with our AAV9 expressing shRNA against BMP22, you see that you can restore a normal nerve conduction velocity even as soon as 1 month of age. So basically 3 weeks after the injection, you already get a normal nerve conduction velocity, and this is maintained for a long time, up to 12 months. This means this is due to the fact that actually the myelin is correctly formed, the myelinating segments are formed correctly, so the nerve conduction velocity increase. And then what we did is check the performance, the motor performance of the animals. So for that, we did first a rotarod test. So I guess all of you know the rotarod test. It's a rotating rod, and the animal has to stay on the rod, and we measure the time that there is fall. Okay, and you see that actually the normal animals, they can stay quite long, 80 seconds on the nerve, in average, of course, on the rotating rod. And you see that the diseased animals, they fall quite fast, and they have less, they stay less long. And when we treat the animal with our vector, you see that they can maintain the normal, almost the correct performance over time, over 12 months. We also measure the strength of the animal, and we do a grip test, and we allow to measure the strength of the line. And you see that the sick, the diseased animals, they have less strength than the wild-type animal in gray. And you see when we treat the animal with the diseased animal with our vector, we allow them to prevent the development of the disease and maintain the force of the animal. We did also measure the sensory aspect of the disease. So we know that the disease, because it destroys the myelin of the nerve, it reduces the pain, the sensibility to pain. Okay. So, for example, the people they feel less pain at the extremities that we do because they have lost their fibers, the myelin or the fibers, and it's the same for the rat, diseased rat. You see, so here we apply weight on the leg of the animals, and we measure when they start to feel pain because they express it. Okay, and you see that the CMT1 animal, you have to apply more weight before they feel pain. They are less sensible for pain. Okay, but when we treat them with our vector, then they recover the sensibility to pain. Okay, and so both the motor and the sensory aspect are the aspect of the disease are prevented by the gene therapy approach. What we do also, because we are planning some kind of one-day clinical trial, and we know that a clinical trial is really dependent of the outcome measure that you have to use to show that the treatment is effective. And one of the outcome measures we wanted to test is human skin biomarkers. So it has been developed by our collaborators in Germany, Michael Sereda and Robert Friedrich, and they have found that a set of 9 biomarkers, both in human and in CMT1 rat, were correlated with the severity of the disease. So we tested whether this marker could be used as outcome measure for our treatment, gene therapy treatment. And we found here, you can see, that actually you can, only with the 3 best biomarkers, you can differentiate the wild-type animal from the sick animals, but also you can differentiate the sick animals that has not been treated with the vector, with the correct AAV9 expressing shRNA, with the animals that have been treated for CMT1A. Meaning that this biomarker could be used as an outcome measure for the therapy if we develop a gene therapy for patient. What we know is also that when you want to do a clinical trial for gene therapy, the 2 main adverse effects that are known for gene therapy is first too broad biodistribution, so meaning the vector goes to too many organs. When you inject it, for example, in the brain, then it can go to the liver, it can go in the blood, in the lung, it can go different places. And if you it can be a problem for the therapy, because it can do off-target effect. So what we did is look at the distribution of the vector when we inject it in the nerves. We found that actually almost all the animals had a gene therapy vector in their nerves. And you see when you measure the amount of vector that is introduced in one cell. So here it's a viral genome per diploid cell genome. So basically the amount of vector you have in one cell. And you see you get 0.5. So you get 1.5 viral genome per cell genome in the nerve. And we know that actually myelin Schwann cell represents 50% of all cells of the nerve. So it means basically, because the vector is really specific for myelin Schwann cell, that you get basically 1 vector copy per cell, myelin Schwann cell. Okay, so you get really high transduction rate in peripheral nerves. But when you look at where the vector goes somewhere else in the body, we look at GHUs, spinal cord, heart, liver, spleen, kidney, and we found that actually very few animals had the vector in other organs. You see, some of them have it in the GHU, in the liver, in the heart, but most of them, actually all of them, none of them has it in the kidney, for example. And when you look at the number of vectors that you have per cell in each of these organs, you see that we have very little. Showing that actually when you inject the AAV9 into nerves, the vector mostly stay in the nerve. It does not go everywhere in the body. And that's really interesting because it can prevent off-target effect. And then the second adverse effect you get when you get gene therapy, it's the immune response that you can generate, that the antibody, the body generate against the vector that you injected. So what we did is test the immune response of the animals against the vector we injected, and then we found that only 1 out of 5, well, actually 2 out of 10 of the animals we tested had an immune response against the vector we injected, which is very low. Meaning that the immune system does not really detect the vector we inject. And although we found that the titer of the inactivating factors that are in the blood, it's actually very low. It is just above the limit of detection. Meaning that when they detect it, when they generate an immune response against the vector, actually the immune response is really weak. Okay, so it means that altogether that actually when you inject the vector in the nerves, peripheral nerve, the vector stays there and does not generate a large immune response. Okay, so I want to thank the people that did the work with in our lab, Benoît Gauthier mostly and Hélène Ajar, but also all the other people that participated. Also our collaborators, both in Patrick Aubourg and lab, and Robert Friedrich lab, and Sylvia Suarez, but also Caroline Le Guiner and Virginie François Le Razanet in Nantes. Thank you very much for your attention.
Thank you.
Yeah, maybe if I have time, I don't know if I have time, but I did a comparison of our approach with the other existing approach for the treatment of CMT1A disease. So we are not the only one developing a gene therapy for CMT1A disease. And there are, for example, well-known treatment in development from Pharnex, which is a mix of 3 different small molecules. And also, Ionis Pharmaceutical is developing a small siRNA that is directly injected in the body without any AAV virus. And Sarepta is also developing an AAV gene therapy, but the treatment is directly, they directly treat the muscles. And they express into the muscle a neurophosphin 3 that is supposed to prevent the degeneration of the axons. Okay, so I compare our treatment with in the same animal, the CMT1A rat, except the last one is a Tremblanger mouse. Okay, and we check what was the most efficient result, and you see that what we get, it's for example, looking at the nerve conduction velocity, we get 100% recovery with a single injection just after birth in 6-month-old rat. While our concurrents, they get 55% in Pharnex, they get 50% recovery. You see here the recovery versus the diseased animals here. But Pharnex, Ionis get a quite nice recovery, while the Sarepta approach is much less efficient. So meaning that our gene therapy is a quite efficient way to treat CMT1A.
Okay, thank you very much, Nicola.
You're welcome.
So I have a question regarding the diffusion of this AAV. So in terms of clinical application, maybe I miss it. If there is a huge diffusion in a PNS, in the nerve of this AAV, the diffusion. Do you hear me? No, the diffusion of this AAV, do you have some data on the diffusion? Do you have a spread infection of Schwann cell all along the nerve, or is it local?
No, what I show, we look for the diffusion of the vector along the nerve. And for example, in rat, we could cover the full sciatic nerve with a single injection, meaning that we can, with a single injection, we could cover 2 to 3 centimeters of nerve. Okay. We did that also, we did check that on non-human primate. And in non-human primate, with a single injection, without optimization of the injection, we can cover up to 5 centimeters of the nerve, of the sciatic nerve. So the diffusion is quite large in nerves.
Yeah, but if you consider the other nerve, so because in I guess in CMT1A, in human, the all nerve are affected, no? And if you think about gene therapy, so do you think about applying different injection or?
No, actually in CMT1A, all nerves are affected, but actually okay. Only the symptoms are due to distal nerves. So the main symptoms in the hand and in the feet are due to distal nerve degeneration. So most of the people, they live very fine with that, except that the distal nerves are degenerating and they have the deformation of the extremities. So it's not necessary to transduce all nerves, because not all nerves are inducing problems. The idea we have is to transduce only the nerves that lead to the symptoms. So in our case, for example, it won't cure the disease. Our idea is not to cure the disease. Our idea is to treat the symptoms. And treating the symptom, we allow the people to have a normal life for a long time. So it's not necessary to treat all nerves.
Okay.
Okay,
Nicola,
we have several questions from the chat.
Yeah, Nicola, I have a question.
Yes.
Nicola, so I have two questions.
You.
Yeah, I have two questions. First, you didn't mention if fibroblasts are in the nerve are infected, or you just mentioned that non-myelinating Schwann cells are not infected. And my second question is, did you try other vectors, and do you have vectors efficiently infected non-myelinated Schwann cells?
So you are right. Actually, what I mentioned is actually we cannot differentiate. I mean, it's difficult to differentiate the non-myelinating Schwann cell from fibroblasts. And actually, we have so little amount of cells that actually we pull them under the same name on non-myelin producing cell.
Okay.
Okay. So I don't know about fibroblasts. Anyway, if they transduce fibroblasts, it will be very little.
Okay.
Okay. So I don't know. It's less than 1%. Okay. So we weren't gonna go into that because it will be really a time killing experiment looking at very small amount of cells. And then we did test AAV9 on AAV10. We could not see, except that AAV10 is less effective transducing myelinating Schwann cell, but is not more effective transducing non-myelinated cells. Okay, it's not working better. We know from the literature that people test from AAV1 to AAV8, and it's the reason why we didn't do, because it was done before. And actually, none of them was really efficient transducing cells in peripheral nerves. So, and actually, not efficient transducing myelinating Schwann cell for sure. And what we saw on the data they presented, that they don't even transduce non-myelinated cell also. So far, no, there is no vector for non-myelinating Schwann cell.
Okay, thank you.
Welcome.
Hello, Nicola. This is Thierry. Can you hear me? Nicola, can you hear me?
Yeah, I hear you.
Yeah, this is Thierry from Bordeaux. I have a question, a very basic question. You've shown that for the CMT1A rat, the segments between two nodes of Ranvier were shorter, and you yes. also showed in the same time that the nerve conduction velocity was slower in this for these rats. Yes. So the basic question is, why is it slower? Because at each node of Ranvier, because it's a saltatory conduction, so each node of Ranvier, the nerve conduction velocity must be more or less the same. So shorter segment does not mean necessarily slower conduction, or maybe I'm wrong. Can you comment on that?
Yes, sure, I can comment. I mean, this has been shown for a long time now, actually, that you get an optimal length for the internodes that allow you to get fast conduction. And if okay, so the longer is the cell, actually the faster will go the action potential, because they have to jump from a longer distance. So actually, the longer the better. It has been shown. For now a long time that shorter segments make a slower nerve conduction velocity. After we try to increase we figure out a way to increase the internodes, to make them longer. But after a while, if they are too long, you have a block, conduction block, because the axon potential cannot jump anymore to the next node because it's too far. But actually, if they cool, it will go even faster. The longer, the faster. Effect when it fails.
Okay. Okay. Thank you, Nicola.
Welcome.
Okay, so we have a question from the chat from Hélène Irbeck. She wants to know if the model develops similarly in males and females.
Is that a question for me?
Yes, that's a question for you.
Okay, sorry, I didn't hear it then.
So the question is, does the model develop similarly in males and females?
Yeah, maybe you write it down. I don't understand the question. I'm deaf, you know, and don't hear everything. Can you write it down?
Yes, I can.
Yeah, go ahead. That's the nice thing with this kind of webinar. I can read too.
Hello?
Yeah, okay.
Oh, maybe I can just ask.
About male and female. Sorry, I answered the question. It's yeah, basically there is no difference between male and female. There is no gender shown. So actually, in our study, we use both male and female, and we could not see a difference. One more question?
So there is one question again from the chat. So you injected this HRNA from PMP22 at 6-7 days in rat, and what is the equivalent age in human if you want to do a clinical trial?
Yeah, so it's a bit far away, but we already have an idea. We plan to inject some. So, well, in human, it's much larger. Okay, and now we did that the trial in rodent. We did one non-human primate. We have to do more non-human primate because actually the nerve of humans are not exactly the same as in rodents. Okay, the nerve actually is collect the fibers into what we name fascicles. Okay, in mice, for example, the sciatic nerve is only one fascicle, but in humans, the sciatic nerve is many fascicles. So actually, the nerve anatomy is not the same, and it's the reason why we have to do more experiment in non-human primate, in macaque, because the macaque nerve anatomy is close to the human nerve anatomy. So we plan to do experiment in macaque. So I can tell you about the dose we plan to inject into macaque. So we plan to inject not the exact dose, of course, not yet, but around 10 to the 12, 10 to the 11, 10 to the 12.
Okay, thank you. So, Laurent, do you want to conclude?
Sorry?
No, I was speaking to Laurent, if she's
Yes, I will conclude now.
Okay.
And so we have now come to the end of this webinar. I would like to thank you all for attending today's webinar with a very special thank you to our lecturer for their excellent presentations. Congratulations and thank you very much. Should you still have any further questions to one of the speakers, I welcome you to submit those to us by email, see here on the slide, and we will get back to you as soon as possible. The recording of this webinar will be made available to anyone interested on our websites within 2 weeks. Thank you and have a very good day.